I'm Giulia Angelini, Research Fellow at Università Cattolica del Sacro Cuore and a member of ESD Study Group on Metabolic Surgery.
Hi, I'm Lydia Castagneto. I'm a Research Fellow at the Department of Surgical Sciences at Sapienza University of Rome. We focus our studies on the mechanisms of prompt resolution of type 2 diabetes and insulin resistance after the bypass of the upper gut in subjects with obesity. Although many medications are available to treat type 2 diabetes, less than 50% of patients achieve adequate glycemic control with medical therapy. Data from several randomized controlled trials have demonstrated that bariatric surgery is more effective than intensive medical therapy for managing type 2 diabetes, and many patients achieve normal glycemia after bariatric surgery without the need for diabetes medications. Bariatric surgical procedures that bypass the upper gastrointestinal tract, such as bilipancreatic diversion and Roux-en-Y gastric bypass, result in greater type 2 diabetes remission rates than those that maintain intestinal continuity. We recently showed that even when subjects with obesity display the same weight reduction, specifically 20% of their baseline weight, whole body insulin sensitivity is improved more after BPD than after Roux-en-Y gastric bypass. Given that the major difference between gastric bypass and BPD derives from the bypass of the jejunum in the latter, it is conceivable that the jejunum secretes factors responsible for insulin resistance onset.
In our latest study published on GATT, we postulated that the jejunum takes a crucial role in the development of insulin resistance, and therefore we assess its role both in humans and experimental animals. In humans, we studied glucose kinetics and insulin secretion before and 1 week after Roux-en-Y gastric bypass or bilipancreatic diversion. While in Roux-en-Y gastric bypass only the duodenum is bypassed, in bilipancreatic diversion the duodenum and the entire jejunum are excluded from food transits. In pigs, we excluded the jejunum from the intestinal continuity and attached its proximal and distal ends to the skin, while the remaining bowel was reconnected with an end-to-end anastomosis. In this way, it was possible to obtain a jejunal loop with its vascular and nerve supplies intact, which could be infused selectively with glucose isotope, while a glucose tracer was infused intravenously to assess the modification of insulin sensitivity. Finally, in vitro studies investigated the role of circulating factors in hepatic gluconeogenesis and muscle insulin signaling. To test the early effect of the jejunal bypass action on the improvement of insulin resistance, we studied 24 subjects undergoing Roux-en-Y gastric bypass of bilipancreatic diversion at baseline and 1 week after surgery. The subjects were matched by age, basal plasma glucose, and insulin. While hepatic insulin sensitivity ameliorated after both types of surgical operation, peripheral insulin sensitivity was increased only after bilipancreatic diversion, and insulin secretion was consensually reduced as a mechanism of compensation for the reduced degree of tissue insulin resistance. In contrast, the total insulin secretion rate in response to the oral glucose challenge was increased 1 week after Roux-en-Y gastric bypass. To prove that the jejunum is central in the onset of insulin resistance, we performed jejunectomy in pigs and made a jejunal loop with intact nerve and vessel connections. We administered a glucose load in the stomach and infused glucose tracers 1 week after jejunectomy, and in a different day the glucose load was injected in a jejunal loop. Infusion of glucose in the jejunal loop worsened insulin sensitivity as compared with intragastric administration following jejunectomy. Metabolomics analysis reveals a similar pattern of expression of several metabolites in both humans and pigs. Indeed, bile acid and proline increased particularly after bilipancreatic diversion and in the jejunectomy experimental condition, while other metabolites like gluconeogenetic substrates as well as branched-chain amino acid decrease more after bilipancreatic diversion than after Roux-en-Y gastric bypass. The mechanism of action of the jejunum in hepatic and peripheral insulin resistance was confirmed by our in vitro studies. We found that plasma obtained during glucose stimulation of the jejunum in pigs and during oral glucose tolerance test preceding surgery increased phosphenol pyruvate carboxykinase and glucose 6-phosphatase mRNA expression in primary hepatocytes cultures. This suggests that the plasma contained substance counteracting the action of insulin and stimulated the rate-limiting enzyme for gluconeogenesis. Therefore, this enzyme facilitates cataplerosis to generate glucose in the Tirivella loop and in humans. Instead, jejunectomy in pigs and jejunal bypass in humans act inversely, amplifying the anti-gluconeogenetic action of insulin. The plasma collected during the glucose stimulation of jejunum in pigs and during the oral glucose tolerance test before surgery inhibit glucose uptake and GLUT4 expression in myoblast. The opposite happened with plasma drawn during intragastric glucose challenge in pigs after jejunectomy and in humans after jejunal bypass.
In conclusion, we show that the jejunum plays a major role in inducing insulin resistance, and then its bypass ameliorates insulin sensitivity. In clinical practice, pilopancreatic diversion may be superior to Roux-en-Y gastric bypass with respect to improving glycemic control in patients with marginal endocrine pancreatic function at baseline, as is not reliant on boosting insulin secretion. Our results lay the foundation for the discovery of gut molecular mechanism implicated in the pathophysiology of insulin resistance, and thus the new possible drugs for the treatment of type 2 diabetes, mimicking the effect of metabolic surgery.